All right, let's go ahead and get going. My name is Brendan Lucy. I'm the Vice President of Business Development here at Gemini Bio. Today's webinar we're very excited about is the advanced cell preservation, both cryo and non-cryo, with Petaka G3. I get the next slide.
Yes.
There we go. Oh, that's interesting. Got your background now. All right, so anyway, so Gemini Bio has been around for about 35 years supporting cell culture and stem cell projects, serving customers on all 4 continents. We produce reagents used for research, clinical and commercial applications. One of the big exciting pieces of news about us is that we are finishing up our brand new cGMP manufacturing space. Planning on moving in next month, so that's going really well. One of the other big new things we're working on right now in this very scary time, we are working to help improve our ability as a country to detect COVID-19 by providing viral transport media. And as an important note, we are, beyond having someone at my door right now, this is the fun of this time right now. We've got we are Celardia's U.S. distributor for the Petaka G3 we'll be talking about. We'll go ahead to the next one. So just a quick, very broad overview of what we do. Sera and supplements, FBS, human serum is a big part of what we do. Cell culture prep, as you mentioned, the GMP manufacturing of media and buffers. We do raw material dossier consulting, which is a whole other topic we'll be talking about. Another topic we'll be having webinars about soon is our MoxyGo 2 QC instrument that does both cell size, count, and some protein markers so you can tell what's being expressed. Pretty exciting product we're very proud of. And then today's topic are the Petaka G3. On to the next one. And as a reminder, coming tomorrow, we've got our 3rd of these Petaka G3 topical webinars is tomorrow at the same time. And tomorrow we're talking about hypoxic and physiologic oxygen control and monitoring with the Petaka G3. So we're really looking forward to that. And now I want to go ahead and introduce Aaron Haubner. He is currently the general manager at Celardia. With over 24 years of live experience with a background in pharmacology, drug discovery, cell culture lab work, and medical device management. Aaron, thanks so much for being here today. We're really looking forward to this talk and digging into this because this is some pretty exciting stuff. So go ahead and start. Thank you.
Yeah, wonderful. Thanks for the introduction. And thank you, everyone, for dialing in. I really appreciate it. And I appreciate Jim and I for giving me the opportunity to talk about what I think is a really innovative product. You hear that term thrown around a lot, and it's not always the case. I think we have something really special here. It's kind of a game changer in a lot of ways, which makes it a little difficult sometimes to put into a new process. But I think once it's like anything new, there's challenges with change, but then it pays off if it's the right fit. So I'd like to tell you about our flagship product. My company, Celardia, and we make the Petaka G3. That's the flagship product. We also have different versions of the Petaka and accessories. And yeah, like Brendan said just recently, Jim and I, we had a great partnership, I think, set up. And they're going to be the U.S. distributor for us and taking the lead in that. So it's wonderful. And we appreciate the opportunity again. So today's talk is going to, so there's a lot of features and benefits with the Petaka. It does things that no other technology can do, and it does a lot of things that no other technology can do. This is kind of like the feature and benefit menu, if you will. There's a lot of aspects of the Petaka. I could fill a 90-minute talk just going over really meaty data, like just getting into the nuts and bolts of it, about what it does, and you're talking about a whole bunch of different things. Today we're going to focus on cell preservation, and for us that really is, you can divide that up into live cell shipping and then cryopreservation, an alternative form of cryopreservation that I'm going to review for you. It's kind of new and interesting. You can't talk about the Petaka without discussing its unique design and it's closed, slim, efficient, the design and the functionality associated with it. So we're going to talk about the core technology and the IP behind the Petaka and what makes it different, and then how that translates into cell preservation and why that might matter to you. Okay, so I go into the basics. You know, on its face, it looks like a little plastic cell culture disposable. It looks different, but you know, on the outside, it's rather unremarkable. It has all the features and typical features and design characteristics you would expect and you're used to when dealing with plates, dishes, and flasks. It's closed, sterile, single use. It's all polystyrene. It's all class 6 animal-free materials. It's endotoxin tested. You can grow suspension or adherent cells. We gamma sterilize 10 to the -6. That's little differentiation. Some of the companies still go 10 to the -3. But it's a little different when you look at the entry and how you operate it. So everything goes into a closed liquid transfer through a sterile silicone port. And then, of course, we feature our core technology, which is the automatic control of dissolved oxygen and CO2. So we think of it as like a little miniature incubator inside itself. So in that regard, it's often something very different than you get with any other open system. And I'm going to get into that. The cell culture basics, there's really nothing different you have to do with the Petaka that you do with your plates, dishes, and flasks in terms of handling and manual control. If you can fill a flask, you can fill the Petaka. You don't, in that regard, we're a little bit different because other systems, proprietary spinning flasks and these wheels and these other benchtop bioreactors, even the hyper flask, they have these special filling tubes and protocols. With us, it's just filled just like you would fill with a serological. The port accepts pretty much any liquid transfer device, whether it's syringes, micropipetters, repeater pipettes, even serologicals with some adapter tips go right into this. The port here, you can see it's manual. I prefer filling with a 30 cc syringe. It's really hygienic, really closed. You can sterilize the port. You can disinfect it with isopropanol, ethanol, benzalkonium chloride, any hexachlorine solutions, any of the typical disinfectants. So you really, really get a clean, hygienic handling, particularly knowing when it's going in and out of incubators, okay? Every cell type you can ever imagine has been cultured in the Petaka over the last 10 or 11 years now. Primary cells, immortal cells, transfected cells, every kind of stem cell you can imagine, embryonic, iPSCs, MSCs, they all do very well. Hybridomas, everything's been grown successfully in the Petaka. So it has a track record. The core, and this is the core of the technology, and this is what allows really the cell preservation applications I'm going to discuss today. And that is the idea that we control the microenvironment inside of the Petaka. So it's what the cells experience. So typically you have a flask or a dish inside an incubator. It's 21% oxygen in the air. That's the equivalent of about 150 millimeters of mercury of oxygen in the air. But cells don't live at 150 millimeters of mercury. If you can see from the chart, this very common data you've seen, dissolved oxygen to oxygen partial pressure, the PO2, of all kinds of tissues. And you can see typically it ranges in the body anywhere from 25 up to 50. So that's a typical range here, and it's represented by these bar charts. That's where you want to get to, and that's why so many people have glove boxes and oxygen control boxes and tri-gas incubators. We get there with the Petaka, and it delivers it that way. And you would say, well, how does that work? Well, all of the open systems, plates, dishes, and flasks, there's no control of the oxygen. So as the cells consume oxygen, it's replaced at the saturation limit of the oxygen in the air. So it's a complete massive flux in, and the cells are never going to consume more oxygen that can dissolve into the media. And the Petaka is a little bit different, and that goes to the design. So it's not immediately open to the atmosphere. The gas that can dissolve and go into the media in the Petaka has to go around the frame in a closed little microchannel, and it wraps around the frame multiple times, such that this little frame contains and houses within it about 1 meter of microchannel, and it's very tiny. So as the cells consume oxygen, the dissolved oxygen drops. Now, as it keeps dropping, it doesn't go to ultra hypoxia and then your cells die. We regulate it, and the cells auto-regulate the oxygen. Here's a typical profile. So if you're starting with saturated liquid media, the equivalent of 150 millimeters of mercury, if you seed with just a minimum number of cells, like 2 million, you'll see the oxygen consumption brings the oxygen level down until you reach equilibrium. Now, that same equilibrium will be reached. It's just a question of time. So if you see a lot of cells, the drop is rapid. So the cells consume their oxygen. At the same time, they produce their own CO2. And the Petaka is tuned fortuitously, the equivalent of the microchannel design that gets you physiologic oxygen also automatically maintains between 5 and 7% dissolved CO2. So it's really an ideal system for maintaining cells at a really, really nice pH and a physiologic oxygen level without any of the external apparatus. Now, there's a whole lot to that, and we're going to have a webinar tomorrow on bioprocess monitoring, because you can do that in the Petaka in a way you can't do in any other system, and sophisticated, oxygen hypoxia, physiologic oxygen experiments that you can do in the Petaka. We're going to go in more detail that tomorrow. Today, we're talking about cell preservation, why the design of the Petaka allows something different. So I'm going to move on. Is a key feature of the Petaka. It's functionally closed. So once the cells are in Petaka, you don't get any evaporation. Even if you leave it out for days, it's a week, it's really trivial evaporation rate. So because the gas exchange is so restricted. But in a flask or a dish, eat just a few days out in the open, outside of the saturated incubator, you get a lot of evaporation. That's one feature of the Petaka. You don't need a water tray. You don't need humidity in your incubator. Just put the cells in the Petaka into your incubator and you're fine. Well, that has implications for really a lot of implications in terms of lab hygiene and contamination risk. But we're talking today about cell pausing and cell shipping. So I'm going to move on to that. So there's a big push across many industries and applications where they're talking about alternatives to cryopreservation and cold chain logistics. It's been estimated multiple times that cryopreservation cold chain logistics for a variety of cell therapy products may constitute 25 to 30% of the cost of goods once they're producing at scale. And you can't really ship cells or store them for more than, say, 2 to 3 days when they're live in the original media. So there are a number of companies that do this. They are really limited to overnight. The biggest company that I know that does it, it really has a limit of, they're pushing the limit at 3-day. After that, they get their live cell shipping solution, if you call it that. They get a huge return. They get a huge reship rate if for any reason the package goes to 3 days. So people go to cryopreservation, but cryopreservation is complex. It's expensive. You have other facilities. You have more GMP processes you have to open and maintain. The cryo process causes a lot of cell damage, death. We know that DMSO is toxic if it's a clinical trial product, or if you're intending to go that way. Not only do you have to ship it, you have to ship it now to, you have to administer maybe at a center of excellence where they can do kind of a wash before it goes in. And that's a whole nother process there. So it's all adding to the cost. And right now, no technology reliably stores cells really for more than that 3-day limit, except for the Petaka. And that's sort of the introduction to this portion of the talk. So how can the Petaka provide 7 to 10-day live cell preservation at ambient temperature in the original media when there's really no other technology that can do that. And that's because the design allows us to take advantage of a natural phenomenon called cell pausing. And there's a really excellent review by Robinson, came out in 2014, and it just puts it all into perspective in one paper. It's very nice. You can, if you request it, I'll send you a copy. But there's this trick that if the cells are at mild hypothermia, really anything below, say, 32 degrees, and you have low levels of oxygen, physiologic oxygen, the cells go to sleep. And if you can maintain no dehydration, which is not really possible in any of the other open containers, you can maintain the cells and maintain the pH at in a dormant state. The cells simply quit dividing. Their doubling time goes exponential, and they just kind of sit there in a senescent state, not senescent, but a dormant state. And then when you return them to 37 degrees, they simply wake up, for lack of a better term. And this is what the cells look like. Really a very large amount of our customers actually use this for cell shipping, whether it's between labs, at academic labs or at private labs, and even commercial facilities that have validated the Petaka in their GMP process for shipping clinical samples, either between clinics or from patient to clinic or patient to lab. This actually makes up quite a bit of our business. And this is what happens. It's simply in vitro dormancy. The cells simply go to here. They're attached cells, for instance. When they're shipped, they get a little rough handling. It's ultra low oxygen. They're very, very quiet. They're not dividing. Typical example here, after a week or two, the cells might loosely detach. That's about all that's going to happen. And then when you warm them back up, they reattach and they look very healthy and they start growing immediately. There's a natural phenomenon, like I said, it's very similar. I compare it to a bear hibernating, or any of the animals hibernate. They just hibernate. Their metabolic rate drops 90% plus. And then when they wake up, it ramps back up again. So we did some extensive testing. We had a partner lab actually did this. What do the cells do? Because we wanted to see, this is a phenomenon that really hadn't been explored 10 years ago. And we were sort of the first to investigate it in the Petaka in this way. Here's your normal cells at the end of day 14. The cells are very healthy, look nice. And we looked at cell markers, and they just stack up in the G2 phase. So they're not actively dividing. They just kind of are arrested at the G2 phase, and they just kind of sit there. And then after a couple days of recovery, you can see the G2 markers dropping because the cells are proliferating again. And you can see the progression with this image from the microscope. There are really no mitochondria. I can explain more on a phone call if you want, if we can do follow-up, but we found there's no mitochondrial effects, and there's no genomic effects. And this was kind of new at that time. And since then, there's been a lot of work in this area. And all of these findings have been sort of discovered and confirmed and reconfirmed in a variety of systems that show that when you put cells to sleep this way, in the laboratory you can do it, but in practical use, there's really no container or apparatus that allows it other than the Petaka. But in the laboratory, they sort of simulate this, and they find over and over again, there's no long-term damage, there's no karyotype defects or genomic defects or mitochondrial effects. And this is the process. You simply take we have a instructional video on the website, it's very nice. You simply overnight incubate the cells, make sure you incubate them overnight, make sure the oxygen's dropped, because if you don't reduce the oxygen, you can drop the temperature, but if the oxygen level is still high, it's still a hyperoxic environment. You get a lot of reactive oxygen species, you get a lot of oxidative stress, and the cells will not do as well. We've done this in the Petaka. But you put your cells in the Petaka at least overnight, or it could be multi-day incubation. You could be expanding adipose stem cells. Some of our customers do that, and they validated it for clinical use. But at some point, you want to ship or temporarily store your cells for your GMP process for later downstream processing. You simply pull them out, and you either put them in a shipper and ship them, or you just pause them in the laboratory. And when you're ready to use them, you just simply bring them back up to 37 degrees. You can ship cells in the Petaka like you ship a book with Amazon. It's just that easy. This is a typical profile. We probably suspended or paused cells for temporary storage or shipping. We've probably done that in 29 or 30 different cell types and cell lines just in our labs, and our customers have done dozens and dozens more. This is a very common profile for a mammalian cell. So at 7 days, your viability in almost every cell line is going to be over 90%. And at 2 weeks, you start getting significant a drop. So we say the sweet spot for Petaka is 7 to 10 days. 7 days, you're never going to have any problems. Some cells are a little more sensitive, and that 10 to 14 day, you could see viability down to 70%. But that's why we say 7 to 10 days. We've never had anyone say there's much of a problem there. So it really is a live cell, ambient temperature alternative to cryopreservation. And there are some sort of solutions out there, additives that you can add, and they're marketed, and most of them are marketed to say, you know, you can preserve the cells for 3 to 4 days. If you look at the formulations, they're really just stripped down basal media, and they're typically, then you put that into a cap tube or something, and all they're trying to do is mimic what we have already achieved in the Petaka, which is low oxygen, and they just cap it off, which they risk deep hypoxia and anoicus and apoptosis. With us, you don't get that risk because you never go that low. We stay in that low physiologic range. And then they have a stripped down media to help sort of artificially suspend the cell division and cell growth in the cells. We do it naturally. The other solutions that are promoted out there we think are a little bit harsher and a little bit artificial. Plus, most of them really only support, they'll only get behind, say, 2 to 3 day shipping or pausing. We're confident you're going to be able to validate cell pausing for up to 7 to 10 days. Okay. Now we realize not every process only needs a week or 2 of cell pausing or shipping. For longer term storage, you want to go cryo. Okay, so the old method is still in use. Everyone's using cryo vials. It's reliable. It's worked for a long time, but it has its problems. Okay, the current problems with cryo freezing is you're putting cells in a big volume in a small space. You put them in 10%, 12% DMSO. You have cell death upon thawing. People are still thawing them in water baths. When you're done thawing, you have to transfer them into conical tubes. You have to spin them down and rinse them. And then you plate the cells. It's a long wake-up period. I know that when I was doing it, you typically wait 2-3 days until your cells really get going. Depends on the cell type. And then it's expensive to store and it's expensive to ship. You have to ship a big box full of dry ice, and then you have to do it overnight. Not so with the Petaka. But anyway, back to the cryopreservation solution. What's the ideal solution? Well, if you could eliminate trypsinization, because that damages the cells prior to freezing, eliminate excess DMSO, you can eliminate hard to retrieve and identify cryotubes. Everyone who's worked with cryotubes knows it doesn't matter how well you label your box, it can be a challenge. Reading the markers or the labels. You really want to eliminate water baths or holding the tubes under running water. Cell death during the long thaws. I know everyone says, well, thaw your tubes in under a minute. I don't think I've ever thawed a tube in under a minute. It typically takes closer to 2 minutes, depending on the temperature of the water. And you eliminate the long wake-up period and transfer. So we think it's more hygienic. These problems are really innate. It's a consequence of using a tube container. Because if you look at the surface area to volume, it's very, very low in a tube. So you have all the cells jammed in here. And if you freeze, you're going to freeze from the outside in. It's going to be a very slow process because, well, you have a very low surface area for the volume. This is the Petaka solution. It looks like this. So you have the cells attached. You leave a little cryofilm, a thin film of freezing media, no more than 10, 12 microns thick. Then you can freeze very rapidly. That change is very, very important, and it makes a big difference. Okay, so we call it thin layer cryopreservation. This is the Petaka process. If you don't want to mess with trypsinization and transfers and spinning it down and thawing, this is what you do. Withdraw the culture media, takes about 10 seconds. Fill the Petaka with your freezing media with your DMSO. Let it sit for about 30 seconds. That's enough. Withdraw the freezing media, which takes another 10 seconds. Place the Petaka into an ultra-low freezer or a vapor phase. That's it. This is what the cells look like. It's a little cartoon. The cells remain attached. They're not damaged. They're not injured. Because you have such a thin film, ice crystal formation is extremely limited. It's a process understood and described as dynamic vitrification. Because you have such a thin layer and it freezes so rapidly, you get a little bit of that glass-like state rather than a crystalline ice formation state. There's not enough time in the process for the ice crystals to propagate in the way and to create the solute disparity and concentrate solutes in the cell and have them dehydrate. You don't need to. Simply freeze them, vitrified, and then when you pull it out to thaw, you don't have to use a water bath or any other unhygienic process whatsoever. You simply take the Petaka, and let me get the picture. Well, you just get warm media, put it in the port, fill the Petaka. As the warm media fills the Petaka, the cells thaw in milliseconds because the freezing is reversed very, very rapidly. And then you have very healthy cells. This is what they look like. So we froze away dozens, maybe hundreds of cells, Petakas full of cells, and then we pull it out every 6 months and test for 5 years. At 5 years, we quit testing. And they all look like this. This is the cells thawed after 1 hour. Believe me, at this point, they're already metabolizing and growing. They're healthy, they're happy, they stayed intact. All their integrin receptors, all their surface proteins are intact and doing well because there was no trypsinization. They don't have to reattach. And there's a picture after 2 hours. The cells do very, very well. They're healthy, they're happy. We have extremely high viability with this process from where we've tested. And it's a nice alternative if you're looking for something different. So this was what we describe as our advantages that we sort of discovered by accident in the Petaka. When you vitrify in the Petaka, you get high cell viability. There's no transfers. I grow, I freeze, I thaw, and I start growing again in the same sterile container. So I don't ever have to worry about that, which, by the way, in the Petaka, I put into the incubator without the water tray, without humidity, without CO2. It's just, or with CO2, even if you have the CO2, it doesn't matter. It doesn't change the pH any. And I don't have to do any wash or rinse. I can take 2 sides of a Petaka, culturing, say, at close to confluence, something like 12 million cells on each side, and then I have less than 1 milliliter of residual freezing solution, so that when I fill it with 25, I have less than 0.4% DMSO in my post-thaw media, which means I don't need a wash. You never have to do a wash. There's no thawing, like I said, no thawing of vials. Cells are ready to go in 1 hour. I call them assay ready. They're ready in 1 hour. And because you can put so much cells per Petaka, the density is not that far off compared to a 9x9 box of cryo vials when you're talking 1.5 million cells per mil. If you're looking at those numbers, the frozen cell density is very comparable. So you can freeze away in high density. The thawing process is very rapid. The freezing process is very rapid. We eliminate the slow cooling, the controlled slow cooling that you have to do. And in our hands, we've gotten excellent, excellent results. So it's an alternative. If anyone's thinking about growing these cells, banking them, thawing them rapidly in a more hygienic way, it's non-traditional, but we think it's better. You have to adjust your process and then ship or store the cells in the Petaka. We think both are very good and very important because, they're alternatives to processes that are known to be slow and create a little bit of a bottleneck, particularly shipping the cells. So we think the Petaka is very special in this regard. And it could be a solution for you if this is something you're thinking about. So we're reaching out to anyone, doing biobanking or preserving cells for clinical trials or scaling up for pilot studies. This might be an alternative for you because it's one platform where you can do it all. Now I'm going to review the Petaka a little bit because it does more than that. And if you're going to miss some of our other webinars or talks, we think it's really good because not only can you freeze and thaw and ship and culture, you can also centrifuge in it. So we eliminate all the transfers. You can do your fluorescent imaging. You can do cell sorting within the Petaka with magnetic beads. You can lift the cells non-enzymatically with these little scraper bars in there. And like I said, cryopreservation, cell shipping, everything's done in the Petaka, and everything is always at physiologic oxygen. You never have to worry about if your incubator's at 5% or if your CO2 runs out. With the Petaka, everything's automatically regulated and automatically managed by the technology in the device. So it's a device, it's a disposable device solution that solves some of these more complex process problems that you deal with on a daily basis. And because of that, we think it's something new and innovative. And if anyone's interested, they can talk to me, they can talk to their Gemini rep. All of the products, all of the Petaka products are available through Gemini, and we encourage you to reach out.
Hi, Aaron. Thank you very much for that. That was great. I see a couple questions, and if any of you have any additional questions, feel free to log in to the Q&A and type them in there directly. The first one is, what about human T cells shipping in this Petaka device, specifically primary T cells?
Yeah.
Anything around that?
Yeah, sure. So I would say about half of our total sales and half of our total volume goes to clinical applications, whether it's regenerative medicine clinics or people growing up some stem cell or cartilage cell that goes into a clinical trial. Without saying who uses it, because I'm not so sure it's everything can be disclosed at this point. Yeah, we know that companies are using it in their T cell trials. We have customers for that. We don't know exactly how they use it, whether they're using it for cell pausing or transport within the facility, or if they're shipping primary samples from the clinic. We don't always get all that level of detail, but we know they're using it for T cell applications, and that's actually multiple customers are using it either in their clinical trial or in a product that's actually on the market today. Let me say that. But yes, there's definitely some experience with the T cells in the Petaka. I don't know if that's a good answer. I'm just feel a little bit restricted in what I can say.
Thank you. We got another question here. How many cells can you freeze on your cryofilm?
Yeah.
In there, yeah.
Yeah, it's so it depends on the cells. It depends on what your cell density is. The interesting aspect of the Petaka is the growth potential density. It's the size of a T75 flask in its surface, but it's double-sided. So every Petaka, it's remarkably small, but you get the equivalent of a T150 flask. So it's 150 square centimeters. So a typical, the range is somewhere between 10 and 15 million cells per 75 square centimeters. So we say somewhere 24 up to 30 million cells per Petaka. So that's what you could expect for an adherent cell.
All right, and I see another, I had a question that I thought might be helpful for people if you talked a little bit about this. Talk about, when people are first incorporating in this, sort of we have that sampler kit and how the beginner kit helps them out and how that all works.
Yeah, sure.
If they wanted to try this, how that would work.
Yeah, sure. Handling the Petaka is actually quite intuitive. We have instructional videos. Just a couple of rules. You hold it vertically because of the design of the air channels. You want to hold it vertically, and then you fill and you aspirate and dispense just like normal. The only real difference is you're going through the port instead of an open lid or a flask lid. which we think is much cleaner and much more hygienic, particularly if you're going to later on scale into transition into clinical process. We think it's good to get on this platform right away. And then what you really need, I mean, the core device is just the Petaka. There's really just a few accessories that you need to get started. I prefer syringes, okay? I think that's the recommended instrument to use for liquid transfers. So you just buy your sterile syringes, and you get the, we sell blunt tips to go into the syringe, so it's very safe. And they work really well with the port. That's our recommendation. And then you just really need some stands, because particularly with adherent cells, you want to stand them up and culture vertically. And then suspended cells culture horizontally. So we sell some we sell some reusable, autoclavable, really rugged silicone stands. You can buy them all separate, or for people to get started, it's a really easy, economical way to buy a basic kit, and I think you guys sell that.
Yep. Well, thank you, Aaron. I think we have answered the questions. If anything else comes up, please keep in mind, just reach out to us at gembio.com. We can put you in touch with Aaron directly and have a person face-to-face webinar, conference call, so we could talk more about any details you have and see how it applies to your specific research, but we'd love to help. I think we're good for now. So thank you all for attending again. And we will be posting this video later today and sharing it with you if you want to reference it or share it with any of your colleagues. Thank you very much from all of us at GenBio and Solartia.
Yeah, thank you.
Have a good one.